Assalamu alaikum, my dear students. Hope you all are in good health and enjoying your online learning. So today's topic is TCA cycle, or it is also known as citric cycle, citric acid cycle, or kreb cycle. TCA means tricarboxylic acid cycle. Since tricarboxylic acid is involved in this cycle, so it is also called as TCA cycle. So TCA cycle essentially involves the oxidation of acetyl-CoA to carbon dioxide in water. And you all are aware that last time we discussed glycolysis of carbohydrates or sugars, and from there, if you remember, the second last step of glycolysis, there was a kreb cycle there, and I told you that we shall discuss this kreb cycle in the next class. So today we're going to discuss that what is actually happening in the kreb cycle. So this is basically a continuation of glycolysis, but it's a new topic also according to your syllabus. If you go through the syllabus outline, this is a new topic under the heading of citric acid cycle. Okay, so this is the same. TCA is a synonym or the other name of kreb cycle. So let's see what will happen in TCA. It's basically a cycle, and the cycle means like there's a series of reactions which are taking place within this cycle, and this is the central metabolic pathway. The TCA cycle or the kreb cycle is the final common oxidative pathway for carbohydrates, fats, and amino acids. Now, the kreb cycle supplies energy and also provides many intermediates required for the synthesis of amino acids, glucose, heme compounds, etc. I think you all are aware that what are heme compounds. They are the iron-containing compounds. We discussed heme compounds or heme molecules when we were learning about the electron transport chain. Yeah. So TCA cycle is the most important central pathway connecting almost all the individual metabolic pathways. So let's discuss what is the definition of kreb cycle or citric acid cycle. So citric acid cycle or TCA cycle or tricarboxylic acid cycle essentially involves the oxidation of acetyl-CoA to carbon dioxide and water. Basically, if you remember, this acetyl-CoA is also a molecule which is formed in the glycolysis. So you have to, just recall the information you have already learned, and you have to put that information into this cycle. So location of the TCA cycle, now where TCA is happening, the reactions of TCA occurs in mitochondrial matrix in close proximity to the ETC. Now, if you remember mitochondria structure, there was an inner layer of mitochondria, the outer layer, and there was in between the two layers there was a matrix. So this TCA or kreb cycle, it occurs in the mitochondrial matrix within close proximity, means very close to the ETC cycle, electron transport chain. Okay, just recall those things which we did in class. What is ETC, where it was happening. So it is happening very close to electron transport chain. So what are the reactions which are involved in kreb cycle? Now let's see. The first reaction is the oxidative decarboxylation of pyruvate to acetyl-CoA by PDH complex. This step is connecting link between glycolysis and kreb cycle or TCA cycle. Step 1. Formation of citrate. Oxaloacetate condenses with acetyl-CoA to form citrate, catalyzed by the enzyme citrate synthase, and the reaction is inhibited by ATP, NADH, citrate competitive inhibitors of oxaloacetate. Step 2 and 3. Citrate is isomerized to isocitrate. So citrate is isomerized by the enzyme econitase. This is achieved in two-stage reaction of dehydration followed by hydration through the formation of an intermediate cis-econitase. Step 4 and 5, formation of alpha-ketoglutarate. Isocitrate dehydrogenase, and the abbreviation form is ICDH, catalyzes the conversion of oxidative decarboxylation of isocitrate to oxalosuccinate and then to alpha-ketoglutarate. The formation of NADH and the liberation of carbon dioxide occurs at this stage. Stimulated by isocitrate, NAD positive, magnesium, ADP, calcium positive 2 ions. And the reaction, this stage is inhibited by NADH and ATP. Step 6, conversion of alpha-ketoglutarate to succinyl-CoA. Now this occurs through oxidative decarboxylation and it is catalyzed by alpha-ketoglutarate dehydrogenase complex. Alpha-ketoglutarate dehydrogenase is an multi-enzyme complex. At this stage of Krebs cycle, second NADH is produced and the second carbon dioxide is liberated. Step 7, formation of succinate. Succinyl-CoA is converted to succinate by an enzyme named as succinate thiokinase. This reaction is coupled with phosphorylation of GDP to GTP. This is a substrate level phosphorylation. Now GTP is converted to ATP by the enzyme nucleoside diphosphate kinase. Step 8, conversion of succinate to fumarate. Succinate is oxidized by succinate dehydrogenase to fumarate. This reaction results in the production of FADH2. Step 9, formation of malate. The enzyme fumarase catalyzes the conversion of fumarate to malate with the addition of water. Step 10, conversion of malate to oxaloacetate. Malate is then oxidized to oxaloacetate by malate dehydrogenase. The third and final synthesis of NADH occurs at this stage. The oxaloacetate is regenerated, which can combine with another molecule of acetyl-CoA and continue the cycle. Okay guys, so I know it's quite complicated. There are so many enzymes which are involved in this cycle. So you have to just memorize the steps, the 10 steps which are involved in the Krebs cycle, and you should be aware about the enzymes, like what enzymes are involved in the conversion of the molecule from one molecule to another molecule, and you should be aware that what are the end products we are getting. Now if you go back to glycolysis, you can see there after the Krebs cycle what we are getting. We got NADH and FADH2, and that these two NADH and FADH2 are going into the electron transport chain. So basically the Krebs cycle gives you NADH and FADH2 which will enter into ETC cycle or ETC. As a result of that we are getting ATP. But within the Krebs cycle there are so many steps, like 10 good steps are involved to to ultimately give you these NADH and FADH2. So you have to memorize these enzymes and the molecules which are converting in the Krebs cycle, and you should be aware of the end product. Okay, so if you see this pictorial form of Krebs cycle or tricarboxylic acid cycle, you can see this pyruvate. Now from where this pyruvate is coming? Yes, you're right, it's coming from glycolysis. Now after what's happening to the pyruvate, it is converting into acetyl-CoA, and from here in the glycolysis metabolic pathway we just write acetyl-CoA is entering into Krebs cycle or TCA cycle. Now here you see it's just like a crab, like as you know that crabs have many claws and many legs, so you can see there are many steps involved in the Krebs cycle. So you can see this is a pictorial form of all the 10 steps which we just discussed. Okay, so you just need to go through all these very carefully, just memorize the compounds which are involved in this Krebs cycle, and try to learn the enzymes. Like you must be aware what are the enzymes which end with ASE, aconitase, here succinate, thiokinase. So these are all the enzymes which are involved in these 10 steps. So I think for today we should stop here. I want you guys to just go through it very carefully, learn the steps, all the 10 steps you should be remembering, because this Krebs cycle will be involved in other metabolic reactions also. So just memorize it, go through it very carefully, and inshallah we shall discuss it further in the next class. Hope this lecture is clear to you, and inshallah it will it will continue in the next session also. So once it is finished, then we will have a question answer session also, inshallah. Okay, thank you very much for your kind attention, and Allah Hafiz.